All right, so next we want to take a look at some of the structures of bacteria. And we're going to look at, starting with those around the outside. I just want to point out this figure here real quick. Figure 4.1 in your text is an overview figure, and it's going to be a good place to go back and summarize each of the different things we talked about. Okay, we're going to talk about most of these bullet points here. So refer back to that when you want to reference each one of those. It doesn't have a figure number, but page 82, this diagram here is just showing a layering system. So this is showing from order of what's on the outside moving towards the inside, and then in the middle of the cell, the internal part here. So this isn't really all that important, but if you like kind of a visual diagram of how things are actually layered, that's a good place to go. Okay, so we're going to start around the outside of the cell, and what are referred to as the external structures, also referred to as appendages. So in a microbial standpoint, an appendage is something that is usually used to help a bacteria do one of two things, and that's going to either be to move around or to attach, so motility or attachment. So typically you have one of those two purposes being met, and a handful of ways in which that's done. So these appendages serve these two functions in a handful of ways. As it says down here, what I talk about is the most common forms, not necessarily all-inclusive. So the first is considered a flagella. Flagella, this goes back, I'm up plural and singular, flagella. This is actually a plural form, and flagellum would be a singular. So, I think, there we go. Oh, I said it right there. I'm sorry, I didn't realize I had that. Anyway, the flagellum or the flagella is, it can be thought of like a tail, and it's got a whip-like motion that acts as a propulsion device, basically. Show you some figures here in just a second. Just a few notes here about the frequency and the occurrence of it. So when it comes to the spirilla, the spiral shape, they all have a flagella. Bacilli, it's about 50-50, and with the cocci, only a small handful have these. So again, this is what we talk about here is kind of a generality, and then if you can always look at the details and how they occur within each type. We're not going to focus on that as much. We're just going to focus on what they are, and not so much as to how they occur with other characteristics like we did with the arrangement. So we're just going to kind of look at them as a separate thing. Real quick, let's get some visuals. Flagella is really quite simple in its function and even in its appearance for the most part. There's a couple different types. We're going to come back to these here in just a second and look at the terminology. But here's an example of a simple flagella. You've got the bacteria. This is a bacilli, and you've got a tail extending from the backside of that bacilli there. So that's kind of like your classic view of what that would look like in terms of kind of how you might visualize a tail on a bacteria. There's a couple other different ways in which those are found, and this is refers to as the flagella arrangement. So let's go back to the PowerPoint just real quick. On the notes there, it asks you to draw the four flagella arrangements. It says it right here. Draw the four common flagella arrangements. So we want to refer back to this figure, and I'll just do it with you real quick here on the paint document. But the four here, well, let's just do it over here. So we're going to have four different types. So let's draw four different bacilli, bacterial cells. Okay. Now one of them, we're going to have a single tail coming out like that. Let's make that a little thicker. Okay. With another one, you're going to have, you know what, I should have moved this one down. Sorry. Because with this one, you're going to have tails on both ends, like that. The third one, you're going to show a cluster of tails, all coming out of one end, like this. Doesn't necessarily doesn't say how many. It doesn't always specify how many, but about 4 or 5 there coming out of one end. And then with this last one here, on the last one, we're going to show tails coming out all around the microbe, all around the bacteria. So all sides like that. Okay, so these are the 4 basic, what we call arrangements. In other words, you can have one on just one side, you can have a tail on both sides, you can have a cluster of tails on one side, or you can have tails all around the outside. Now you might ask, well, could you not have 2 clusters on each, or a cluster on each side, kind of like this one? It's theoretically possible, but apparently it's not seen in nature. And if it is, it hasn't ever been classified. So apparently that just doesn't occur. And it's one of those that maybe just no one's ever discovered it. So this first one here is called monotrichous. So that's what we would call this type of flagella arrangement, monotrichous. So mono, the first part means one, everyone knows that, and then trichous is in reference to tail. Once again, I believe these are sort of Latin or Greek derivative here. This next one here is called an amphitrichous. Make sure I got the spelling right on that. Yeah, amphitrichous. Amphi means something about both. So this is somehow in reference to a tail on both ends, something like that. This third one here is called lophotrichous. That's the clustering on one end. Lopho somehow translates to cluster. That's what you're seeing here. And then the last one is called peritrichous. And peri means around. So around, tails around the cell. So mono, amphi, lopho, and peritrichous. So once again, be able to identify these. You might be asked to spell them. You know what? I just realized I misspelled one of those. Huh. No, I didn't. Sorry. Make sure that you're able to recognize and spell these terms, and then make sure that you can even make a drawing of them yourself if asked to. I might say draw monotrichous flagella or something like that. So, okay. Okay, 3, 2, 1. Okay, so next I'm going to talk just real quick about how the flagella works in reference to the cell itself. I've got just a embedded YouTube video here that just shows it in motion. I'll play that here in just a second. But without going into too much detail, this is just a simple diagram to show how the flagella actually connects to the bacteria, and just a quick demonstration, a quick visual that is on how it actually works. Okay, I'm not going to ask you anything real complicated later. You want to be familiar with what the 3 components are and how those would kind of fit into this diagram here. Recognize where each one goes. So what you basically have with the tail is, let me just add something real quick here. The flagella is one of the most primitive types of motility features in any type of organism. Bacteria are one of the simplest types of organisms, and a flagella is one of the most simple types of motility features that go along with that. So you're looking at a very kind of archaic form of motility for a living organism. Okay, it's a very, very sort of like simplistic way that it works for a living thing. Now the truth is, nothing's really simple when it comes to living things, but in terms of the relative complexity, this is a relatively simple way in which a group of molecules work together to provide motility. So real quick, the components, there's three major components. You have what's called the filament, which is really the major component that at least you think of, which is the tail itself. And that's really what we were drawing there, is how the kind of the filaments extend out. But really that's the tail part itself, okay? Another key part is called the hook. And the hook is this kind of angled component that attaches the filament to the third component, which is called the basal body. Okay, so the basal body is a series of rings that are embedded in the membrane of the cell. It's actually called the cell envelope, and we'll talk about that here shortly. So it's embedded here in the cell, and it's a series of these rings. And what happens is the bacteria can activate mechanisms that will rotate the rings, and as those rings rotate, it'll actually cause the hook to move and the tail to flip and kind of move in different directions like that. So by moving the rings, it's like a steering wheel. You can move the rings clockwise or counterclockwise, and depending on that exact movement, you get motion with the filament here. So let's look at this real quick. So here's going into the cell, and here's this is part of the basal body, and this is rotating here, and that is driving the hook, which then connects to the filament and allows that tail to move there. So now that you've seen it from the inside, go back one more time. So you can see how that tail is moving around, flipping around. Here's the hook there, and it's connecting to the basal body, which is the series of rings that rotate. Oops, sorry about that. Kind of cool, because that is the simplest example of a motility mechanism, and as you can see, it's fairly complicated. And in fact, there's actually an interesting debate here about origins of life when it comes to the flagella. If you look up what's called the irreducible complexity argument, it's a part of a debate in creationists versus evolutionists, and they talk about the bacterial flagellum as part of that. And if that's something that interests you, you can look that up. It's called the irreducible complexity argument, and it mentions a lot of things, but one of the things that's mentioned is the bacterial flagella. So kind of a different context for that same topic there. Okay, the periplasmic flagella is another kind of different version of a flagella altogether. And so it's not mentioned as part of the typical flagellar arrangements, because it's really a different type of flagella as opposed to a different way it's arranged. So it's referred to as a periplasmic flagella, and this is the flagella that's going to be found in spirochete bacteria. So if you go back here to this figure real quick, recall we talked about the spirochetes here. I mentioned that they have a special flagella embedded, and this is, honestly, it's actually called two different things. It's called the axial filament here, and I think here I'm calling it the periplasmic. It's actually called both. So you want to recognize it both ways. Periplasmic flagella, also called the axial filament. It's one of those terms where you have two terms for the same thing. But anyway, this is a type of flagella that is embedded in the membrane. Here you can see a slightly different view of that. This is how it's embedded here. And what it does is it allows the bacteria, as I mentioned earlier, to kind of contort that tail. It's not really even a tail at this point. It's really only called a flagella because it has a similar makeup, similar type of molecule that goes along into the construction of it. But otherwise, it has a totally different function, really. And what it does is it acts as a contortion type device, and it can literally allow the cell to rotate and twist itself. So it's why it's really not classified along with the other ones, because it's a really totally different type of function, only really similar in the type of molecules that makes up that particular thing. So that's the periplasmic flagella, or the axial filament. Okay, so we're looking there at, as a recap, we're just looking at flagella, which are really motility devices, motility appendages. So we're looking really at how bacteria move, and that takes place more or less with one thing, which is a flagella. A couple different types, But that is pretty much the only way that bacteria can self-propel themselves through the environment. Obviously, they can move with water currents and wind and other animals and things like that. So it's not that they can't get around other ways, but as far as them intentionally propelling themselves from one place to another without some outside force, that's really one of the few ways, pretty much the only way that they're able to do that, at least as individual cells. They can grow and spread out their colonies over long distances, but they can't necessarily move one cell. One cell itself can't necessarily move from one place to another. Okay, so the next part here, we're looking at appendages for attachment. So we said that motility is one key thing, one key feature. Attachment is another key feature. Now here, the first thing you might notice is that we got the flagella listed again. Then we've got two other terms here that we haven't seen yet I'll talk about. The flagella is listed here because it can serve a dual purpose, which is for attachment. So it can be used for motility or movement, which is really its primary purpose. But it can then also, in other cases, serve a secondary purpose, which is to help it attach to things. So just recognize that the primary is movement, but a secondary kind of alternative function is for attachment. It can kind of loop around things and help bacteria stick to different things or to each other even. So that's why it's listed in both places, and you'll want to recognize what the primary and secondary function of a flagella is. Now, these next two here are called a fimbriae, and then a pilus, or a pili, plural. Pilus, also pronounced sometimes a pilus. It depends on who you ask. It's like potato. I usually call it a pilus, but it's also pronounced pilus or a pili for plural. So, three things for attachment: flagella, fimbriae, and a pilus. So we talked about the flagella. Only thing you need to know there is that they can also help bacteria stick together or stick to other things, okay? And we're not going to go into anything further about that. Now, fimbriae, we haven't talked about them yet. So what we just want to understand is what they are, and what they are these little bristle-like fibers that stick out from a bacterial cell. I like to think of them as sticktites, or sometimes called cockleburls. Let's see if we can find these here. Sticktites are really like a seed. You might probably have picked these up if you're out walking around in a field somewhere. There's a good picture of them. They're these little seeds that stick to your clothes, usually to your socks, and kind of annoying. But why I mention that is because I think that they're very similar, at least in kind of look, to the fimbriae here. Fimbriae have these little bristle-like fibers that stick out from the bacterial cell, and just like those stick-tight seeds, they allow them to stick to things. Now, the nature of the stickiness is a little different, but the function is very similar. So fimbriae, think of them like the stick-tights. They allow bacteria to stick to different surfaces based on that kind of attractiveness of these little bristle-like hairs here. In a lot of cases, what the fimbriae does is it allows them to stick to kind of a moist surface. Like, for example, in a lot of cases, these were key mechanisms for bacteria that attach to human beings, both good and bad. It's not always bad. But like, for example, E. coli that we find in our intestinal tract, it's going to stick to our intestinal lining, as you can see here in this figure. It actually sticks to the intestinal lining, the intestinal microvilli, with these little hairs, these little fiber-like hairs. And then further, it allows them to stick to each other, kind of like you see here. So this is not only a key aspect of how one cell could stick to somewhere in the environment, but it's also a key part of how bacteria start to form larger complexes like colonies and even biofilms. And I mentioned biofilms in the first chapter, but we'll talk about that. We'll talk about that more, but biofilm is basically when bacteria grow into a large group complex. And that's something that oftentimes begins with them sticking to some kind of surface and then growing from that surface. So that's a key part of that. The last type is called a pili, and sometimes this is referred to as a conjugation pili. I'll explain why that is here in just a second. So we can call it a pili on its own, which again, that's the plural form. Or we can also refer to it as a conjugation pili or conjugation pilus. Either way is technically correct, but why it's sometimes referred to as a conjugation pilus as opposed to just the pilus is because really it implies the primary function of the pilus, which is conjugation. So conjugation is a process by which bacteria are going to swap DNA back and forth. And that's something that we'll talk about in more detail in the next part of chapter 4. So we have kind of two things going on here. One is that we have an attachment mechanism. That's called the pilus. Okay? So this is along the lines of our topic here, which is how bacteria attach to things, attachment appendage. So the appendage is called the pilus. And ultimately what it does is it allows two bacteria to link up to each other and stick to each other in this very kind of specific way. So that's the structural part of it here. And that's the term pilus or pili, in this case is plural, refers to that process of this structure called the pilus linking up and attaching to another bacteria like you see here, or even multiple in tandem like this. Conjugation is a separate process in which some of the DNA from one cell is going to be passed into the cell of another bacteria. So the pilus, the pili, is a attachment mechanism that primarily allows bacteria to undergo conjugation, which is the exchange of DNA between one cell and another. Now, I realize that probably doesn't make a whole lot of sense right off the bat. So I'm not asking you to write an essay on this right now, just kind of throwing out some basic things there. And like I said, we'll try and explain this better in the next part when we talk about that in more detail. So as far as what you want to know here, know what the pilus is. It's an attachment structure. And then know that conjugation, just know that it's the transfer of DNA from one cell to another. It doesn't have to make sense yet. It's not going to right now. But as we go further and further, hopefully that'll all make sense as we go here. It's one of the things in biology you have to just kind of go through layers, and they don't always make sense at first. Hopefully if we do it right, hopefully it'll make sense in the end. But that's part of the process here. So I know it doesn't make a lot of sense. But anyway, that's the third major type of attachment appendage there. So you've got three ways for bacteria to attach. One of them comes from the flagella, which has a primary function of motility. Fimbriae are the bristle-like fibers, and the pilus is the mechanism for other bacteria to attach to each other. And they do that for as a major primary part of that is for genetic transfer. Okay, which is called conjugation. Okay, this next part here, we're going to take a look at what are referred to as surface coatings, and these are structures that occur just above the membrane of the cell, and that's what we're going to get to next, called the cell envelope. So let's just go back and get a visual reference here real quick. I referred to this figure earlier. So we looked at the flagella, we looked at some fimbriae. These are all things sticking out of the cell. They don't have a conjugation. Oh, there's a pilus right there. So these are all things that kind of stick out. Next, we're going to look at something that's still kind of on the outside. It's still kind of an external structure, but this is more of a surface coating as opposed to an appendage. And so we're looking in kind of this area of the cell right here. So this, where's the arrow pointing to it here? Capsule. This right here is called the capsule, the tan coating. That's an example of one of the things we'll find in this topic here. Also, an S layer is shown right here, be just below a capsule, and a kind of an extra layer around the cell. The next part we're going to talk about are the light pink, the darker pink, and the yellow layers here. Those are what are called the cell envelope, and we'll talk about those next. So far we've talked about the very outer things. Next we're talking about what's just around the outside, kind of hugging the cell tight, the surface coatings. And then next we'll talk about the layers that are in between the outside, and then finally the inside will come at the very end. Oh, and if you're looking here, this is your figure showing how the layers would go. So we're right here. Okay, you have really two types of surface coatings. I showed you both of them there. So one is called an S layer. The other is called a glycocalyx, a glycocalyx. I'm honestly not sure what the calyx part means. Glyco is in reference to a sugar, which is what it's largely made out of, is polysaccharides and sugars and things like that. So, like a lot of things, there's different variations of these. We're talking about them in kind of a general term. As it says down here, some bacteria produce either an S layer or a glycocalyx, and then some produce both. It's also important to say some may not produce either. I think most bacteria will produce one or the other, but I'm not sure that that's a given. I honestly don't know the exceptions to that, to be honest, but I think that it's fair to say some probably don't produce either. Okay, so an S layer is this very thin but sturdy layer that occurs just above the cell envelope, which is again what we're going to talk about next. That cell envelope where those middle three layers, the S layer would occur just above that. So in between the very outer appendages like the flagella, and would be just beneath with the next layer that we're going to talk about called the glycocalyx. So here you can see all the other layers removed, showing just that S layer here. Now, what this can be analogized to is shown here, which is chainmail. And you may be familiar with chainmail, call it chainmail-like. Chainmail is a very thin but sturdy type of protective coating that was used in medieval times because it was relatively light and yet very, very sturdy at the same time. Very, very resistant to penetration. So you can think of it kind of like that. It even looks like that from this figure here. But apparently they produce this when they're under environmental stress, and I guess it's a way to kind of help trap water and prevent certain things from entering and exiting the cell, also from potential damage from the outside hurting them, such as sunlight or UV light or things like that. So the S layer is just a basic protective layer. Another interesting thing I want to point out about this is only in recent years was this even discovered as being part of the bacteria. And part of the reason for that was because it turns out they only produce this under environmental stress. And when we grow them in the lab, where we typically study these microbes, that same type of stress is not always as intense as what you'd find in the environment, or it's certainly not the same. And apparently it's not as common for them to make this layer when they are grown in these artificial lab-like conditions, which is really how a lot of the study takes place. And there's a real issue when it comes to studying things in these kind of isolated environments, and this is something that they're learning more about. The problem with that is that you don't always get an accurate representation as to what happens in reality. It's like when people study something in like a drug in animals and mice, and then they try it in people and it doesn't work the same way. Well, it's not the same thing. You're making a comparison, but you're not exactly studying things as they really are. So this is a problem when it comes to all research, and this right here is kind of a telling example of that, where for decades they were studying these and didn't even know this basic feature existed because of the conditions that they were growing them. So now they're aware of that because studies are now getting better, especially when it comes to microbiology. When you're studying microbes, it's a lot easier, more cost effective, a little more boring to a lot of people, but you get a lot more data. You don't have to worry about animal rights and human rights and all that. So nowadays they're starting to study microbes under more natural conditions, certainly more difficult, which is why they didn't always do that, not as easy as is in the lab, but seeing the benefit of that, they're starting to do that more, and that's uncovering new discoveries and giving us a better in-depth interpretation of how these microbes actually work. But I just want to point that out because it's not only just a feature, but it's also a telling insight to how research works and how that's a little bit problematic when it comes to the lab conditions versus actual Mother Nature. So, but scientists are getting better. Every time they learn something like this, they go, okay, we got to do something different, and they try things different. And so it's not always a bad thing that something like that happens, but it is kind of an insight into how that works. Okay, The next part here are called the Glycocalyx, and this is the plural form here, and I will not make you spell that, I promise. Glycocalyxes, that's your plural form of Glycocalyx. I won't even make you spell the word Glycocalyx, just be able to recognize it. What this basically is a loose secretion, kind of like a slimy secretion around a bacterial cell that gives it kind of this extra cushiony layer of protection. You have two basic types of secretions. One is called a slime layer, the other is called a capsule. The difference is whether that secretion is really thick and tightly attached to the cell, such as a capsule, or whether that secretion is a looser and less tightly bound secretion, like you see here in the slime layer. So the terms here actually apply quite well. The slime layer represents the looser secretion, whereas the capsule represents kind of a tighter, thicker secretion. Both of these help in attachment. They also help in reducing things like water loss, but that's not something we need to worry about here. There's a number of benefits, but as it says here, really, it's another mechanism of attachment. And so we did mention that one earlier because it wasn't an appendage attachment. So don't mean for that to be confusing. I'm certainly not trying to be tricky. Probably could organize this maybe a little better. But to clarify, an attachment or an appendage for attachment would be like the pili or the fimbriate. Those are things that stick out from the cell. So those are mechanisms for attachment in terms of an external appendage. A lot of bacteria may not have those or may not always produce them, or even if they do, they may also produce the Glycocalyx in addition or rather than, and this can act as a different or alternative mechanism for attachment. So in this case, they just kind of stick to something, almost like how you might just get something kind of sticky and rubbery to stick to kind of like a hard surface. That's a terrible analogy, sorry. But you get the idea. You can kind of, something sticky that you can just kind of sticks to other things. But that's basically what we're looking at here. So two basic types, a slime layer and a capsule. So what you want to recognize is that these are both a type of Glycocalyx, and each has a similar but different nature. To clarify, they're similar in what they're loosely made up of, kind of the general nature of the molecules, not the exact types, obviously, but they're made up of what are called polysaccharides. These are complex sugars. That's what that means. Polysaccharide is a complex sugar, like a starch, for example, is a polysaccharide. If you're familiar with the liver, you might be familiar with glycogen. Glycogen's a polysaccharide. That's not what these are made of. Different types of polysaccharides. But anyway, slime layers loose, capsules tight. These are used for attachment. Here's a side view from figure 4.13. This, by the way, is not in the textbook. This is an older figure from an older book. This figure here shows a side-by-side comparison and is the figure in your current textbook. So here on the left, figure A, this is figure 4.13. Figure A is a side view of a slime layer, and figure B is a slide view of a capsule.